In this video, we will begin to explore some of the math used in population genetics. But first, let's get a few terms in place. A polymorphism refers to an observation that there is variation in a trait. Pictured here is an example. The spiders in this photo are all the same species of happy face spider, but differing alleles in the population affect the color and pattern of the happy face on them. Cute. If we think about it at the molecular level, a polymorphism is due to genetic variation that leads to two or more alleles that influence the phenotype. So when we use the term polymorphic to describe a trait, we mean that it's a trait that has at least two versions or alleles in the population. In contrast, a monomorphic trait exists when there is only or predominantly a single allele. Underpinning all of this is genetic variation in populations, and often these are single nucleotide polymorphisms or SNPs, which is just a change in a single base pair. These typically account for most of the variation within populations of organisms, including in humans. In fact, each of us contains thousands of SNPs that differentiates us from everyone else. These random mutations can result in new alleles, and in some cases, these alleles can lead to detrimental phenotypes. Here we see variations in the beta globin gene, which, depending on the mutation, can change or eliminate the function of this crucial gene in our bodies. Okay, so now that we have covered that terminology, let's take a look at two simple equations that we use to measure frequencies of alleles and genotypes in populations. As you can see, these equations are very straightforward, so let's jump straight in. Allele frequency is the total number of copies of a particular allele in the population divided by the total number of alleles in the population. Remember that each organism has two copies of each gene, thus two alleles. Genotype frequency is much the same, with this equaling the number of individuals with a particular genotype in a population divided by the total number of individuals in that population. So let's look at an example of each. For allele frequency, consider this population. Pause this video and read the problem. Okay, so now that so here we have a population of 100 frogs, and the number of each genotype are shown here. Note that each frog in the population contains two alleles, with some being homozygous and some heterozygous. So if we are looking at the frequency of the little g allele, we need to first determine the number of little g's in the population. For our homozygous recessive individual, we have four frogs that have two little g's each. So we take our four frogs and we multiply by 2 to account for our 8 alleles in these four frogs. Next, our heterozygous frogs also have some little g alleles, and those 32 frogs have 32 little g alleles. So we add those in too. This gives us a total of 40 little g alleles out of 200 alleles in the population of 100 frogs. Again, remember that our 100 frogs have two alleles each, thus 200 alleles. So the math becomes 40 divided by 200, which equals 0.2 or 20%. So the little g allele is 20% of all alleles in the population. Genotype frequency is even more straightforward. Consider the same population of frogs where we want to know the genotype frequency of the light green frogs. We simply divide the number of light green frogs, which is 4, by the total number of frogs, which is 100, and we get our frequency of 4%. One important note is that these alleles and genotype frequencies always add up to 1, or close to 1, but never more than 1. In monomorphic genes, the allele frequency of a single gene is equal to or close to 1. And in a polymorphic gene, the sum of all alleles should add up to 1. This is a nice little check of your math because if you work a problem like this and get a number larger than 1, you've done something wrong.